Transcription and
Translation by
Amy and
Shreya Patel.
Transcription is a process of where parts of DNA is copied into RNA. This process occurs in the nucleus of a cell.
RNA polymerase is an enzyme that binds to the promoter, which starts at the Tata box of the DNA strand.
This causes the DNA to unwind and split into two different sections, as shown in the image.
After unwinding, one side acts as a DNA template strand.
When RNA polymerase is moving down the template strand, this process is called elongation of RNA.
During elongation, RNA nucleotides binds to DNA nucleotides with an aid of transcription factors. Transcription factors are proteins that initiate and regulate transcription of genes, which does not include RNA polymerase.
After the nucleotides bind, it is also known as the DNA-RNA hybrid complex.
When termination occurs, it stops the process. Then, the RNA strand that is taken out is known as the pre-mRNA.
There are two caps added onto the ends. The 5' cap is made of phosphates, and a 3' tail is made of adenine and is attached to the mRNA. This happens so that when it is ready to move through the cytoplasm, it does not break. It also prevents other enzymes from degrading it.
The introns are taken out because they are empty codes. Then, exons bind. Spliceosome's main purpose is to take out and bind the introns and exons. This process is called RNA splicing. When exons bind, it creates mRNA.
However, if any of these genes are missing, it will affect the process of anticodons and codons bonding, which will later affect the protein that is created.
Translation is a process in which ribosomes are creating proteins. The process occurs in the cytoplasm, mainly on ribosomes.
In the first step, mRNA is going to leave the nucleus and move into the cytoplasm.
Then, mRNA will bind to the small ribosome, which attaches at the start codon AUG. A codon is a grouping of 3 nucleotides that provide information. The tick marks shown in the image along the mRNA strand are also RNA nucleotides.
Later on, the big ribosome, which contains E site, P site, and A site, come in and attach to the small ribosome.
tRNA will float into the big ribosome, so the sites can allow it to attach to the mRNA in the small ribosome.
The anticodon matches up with the codon to create the specialized protein, because each codon and anticodon have their own code. While the tRNA comes in at the top, amino acids will start to bind to each other by polypeptide bonds. The growing chain is known as elongation of amino acids.
Amino acids are done bonding when one of these, UAG, UAA, or UGA, appear, which is known as stop codons.
Then proteins are created.
As for mutations, if one letter of a codon is missing, it can change the whole coding for the protein.